Let's look at question number 25 from GATE Biotechnology 2017 paper. So the question is, the specific activity of an enzyme in a crude extract of E. coli is 9.5 units per milligram of the protein. The specific activity increased to 68 units per milligram of protein upon ion exchange chromatography. So the fold purification is. Before that, let's look into the definition of enzyme activity. So it is used to measure the total units of activity in a given volume of the solution. So the specific activity is used to follow the increasing purity of an enzyme through several procedural steps at different time points of the process. So another term to know is the molecular activity, and it is used to compare activities of different enzymes, which is also called the turnover number. The standard units of enzyme activity is micromole substrate transformed per minute, where in international units it is represented as Catal, which is a mole of substrate per second. And the specific activity is given by micromole substrate per minute per milligram of the enzyme. So moreover, enzymes could be purified by several methods to study their structure and functions. So they can be purified by several methods of precipitation, electrophoresis, and chromatography. And in the question, the enzyme has been purified by ion exchange chromatography. So it uses a stationary phase, which are ion exchangers that could be either cationic or anionic, and a mobile phase that are buffers of differing concentrations. So the basic principle is that it relies on the attraction between oppositely charged ion exchangers and the protein sample analyte, and could be purified according to the charge of the analyte of interest. Now, fold purification refers to the number of times a protein preparation is enriched for the protein that is being purified. So if a protein has a concentration of 1 nanomole before purification, and the concentration of 10 nanomoles after purification, the fold purification is tenfold. So we have like tenfold of the initial protein concentration that we had before purification. So to calculate the fold purification of an enzyme, we can use the formula specific activity after purification divided by specific activity in the crude extract before purification. So in our question, the specific activity of the extract, which is before purification, is 9.5 units per milligram of protein, and the specific activity after ion exchange chromatography purification is 68 units per milligram of the protein. So applying these values to the formula, we do 68 divided by 9.5, and these units, they're the same, so they get cancelled out. So solving for this, we get a fold purification value which is equal to 7.16. So the fold purification is approximately 7.16. So this means that the specific activity of the enzyme increased by a factor of 7.16 during the ion exchange chromatography purification process. So that answers our question. The specific activity of an enzyme in a crude extract of E. coli is 9.5 units per milligram of the protein, and the specific activity increased to 68 units per milligram of protein upon ion exchange chromatography. So the fold purification is 7.16.